here is very close to the frontal lobe, but we're still seeing the lateral ventricle. And if you look at this view, you can see, and I'll leave this on my desk, you can see a brown region that's kind of bulging into that space. That's the head of the caudate nucleus. All right, so it's changing the contour of the lateral ventricle. This is why we model this stuff with the clay, so you get an idea. We're going to model these structures on Tuesday. Okay? So can I erase this picture now? May I? And I'm going to draw it slightly differently from the inside out.
So then where's the head?
On what? On this picture? The head is on the other side of the board.
Oh, okay.
So I cut behind the head.
Okay.
Okay? So in other words, in this model of the ventricular system, if I cut here, you would see the head, but I've cut back here. When you see the head, you can't see the tail.
Okay, got it.
Okay? So if we can see the tail, then we're beyond the head.
Okay, sorry.
All right, so I put that in as red, so let me change the color of the thalamus. And the thalamus is where we're going to end lecture today. We'll take a break as soon as we're done with this. Actually, so this is the thalamus. We're on the lateral aspect of the thalamus. Okay? This is shown on the PowerPoint lecture, so when you go back and see it at home, you can look up the PowerPoint lectures. On the other side of this would be the third ventricle. Just remember, the thalamus forms the lateral wall of the third ventricle. All right? So if I were to put the thalamus in place here, this is the thalamus. Okay. Now, anterior to the thalamus is where I'm finding the head of my caudate nucleus. But between the caudate nucleus and the thalamus, I have my globus pallidus and putamen. So let me make those out of colors similar to red. I'll make them out of purple and blue. The globus pallidus I'm looking for clay. It's in the other lab room. Aha. Play-Doh is okay. It's not, doesn't work quite as well, but since this is all I have.
Have lots of Play-Doh.
Plus you can eat it.
Eat Play-Doh?
It's non-toxic.
Oh, okay. I was like, maybe that kids eat it.
All right, so thalamus is green. All right, so here's the thalamus. Right here would be the third ventricle. Okay, and I don't have purple, but I have two shades of blue. So now the globus pallidus is a pyramidal shaped structure. How do kids get this stuff out of here? So it's going to have a base, and it's going to have a pointy region. All right, it's kind of shaped like that, and like a pyramid. And the pyramid is pointed towards the thalamus. Okay? So if I were to draw it on here, globus pallidus has this kind of triangular appearance, with the base being out here and the pointed region going towards the thalamus. Okay? But most of it starts anterior to the thalamus. The globus pallidus. And then covering up most of the globus pallidus, but even more anterior to that is going to be the putamen. So that's even more lateral. So the putamen is out here. You guys are lucky I didn't do this for the morning class. Too frustrated by not having my PowerPoint. Now, remember the thalamus is medial to the third ventricle. So these models are kind of expensive, so unfortunately you guys get to make your stuff on Tuesday, just on the tabletop. But here is the thalamus forming the lateral wall of the third, stay there, forming the lateral wall of the third ventricle. Nobody listens to me. I say stay there to cars that are coming into traffic and they just pull right in front of me. All right, so there's the thalamus forming the lateral wall of the third ventricle. And then my globus pallidus is going to be right there. And now my putamen I will make a sandwich, okay? So there's the putamen and the thalamus, and the globus pallidus is between the two, just like you saw here. Putamen, globus pallidus, and thalamus. Okay, they're not actually touching because there's fibers going through. Now, the larger caudate nucleus is red. And so its head is going to cover up almost all of this structure here. And form the head, body, and tail. Right? So the head is big and round like a developing frog. And then I'm going to make the tail. This is the caudate, all right? I'm going to put it on this side first before I put it on the other side. So the head fits into the anterior horn like that, and then the body, and then the tail comes in as the roof. So it forms the lateral wall and then eventually the roof of the anterior horn. And it bulges into that space. So when we open up the sheep brain, that's what we're going to see when we open up the lateral ventricle. We're going to see this head of the caudate nucleus bulging out. Okay? And you can do that on some of our sheep models. All right, look at you, you can stop halfway stuff. So it fits into, and that's what gives the shape to the anterior horn of the lateral ventricle, is that caudate nucleus bulging into its base. And then it becomes the roof of the inferior horn. What is in the floor of the inferior horn? The tail of the caudate is the roof. Look up on the lateral diagram.
Those black things.
The diagram on the side. So the tail of the caudate is in the roof. What forms the floor? What's that coiled purple structure?
Gray matter.
Hippocampus.
Oh.
All right, so then if I put this over on the other side, it's going to fit in there like that.
Oh, yeah.
Wrap around form like in that pattern. Okay?
So it surrounds.
So that's the relationship that we see between the thalamus, hypothalamus. So here again is thalamus. Here's globus pallidus, putamen, and wrapping around them in this C shape is the caudate nucleus. Okay? So these structures are involved with the midbrain, and a region of the midbrain known as the substantia nigra. Remember the melanocytes of the skin? And what amino acid did they process to synthesize melanin, the pigment melanin? But remember the amino acid? Tyrosine. And at the time I said it was the same amino acid that our brain uses to synthesize the neurotransmitter dopamine. The melanocytes are related to the same cells that become nerve cells. So this area of the brainstem that makes dopamine makes, as a side product, the pigment. And so it's called the substantia nigra, or the black stuff. And so these neurons making dopamine go up into the brain and function in the regulation of the activity of the basal nuclei. So that's an early stage in Parkinson's disease, is the absence of dopamine. And Parkinson's disease is one of the pathologies of not having the basal nuclei work. So if you look on page 74, the globus pallidus, all of these suppress unwanted movement, all right, so, but they have individual functions. So I read an article about 12 years ago about a surgery that was developed. A gentleman who was a member of the Atlantic Symphony was developing early stages of Parkinson's, and he could move and walk okay, but he had a tremor in his little finger, and he played the cello. Well, you can imagine if your little finger is constantly vibrating that it's kind of hard to push on the strings to create the different notes on a stringed instrument. And so he had a surgery where they screwed his head into a halo to hold it firm and used computers and CAT scans and MRIs to access the image of his globus pallidus, and then a laser knife to go in and zap a region of his globus pallidus that stopped his tremoring of that little finger. Right? Other signs of Parkinson's disease can be rigid little facial expression. When they walk, it's very kind of shuffling, because there's so much descending motor information that both flexors and extensors are contracting at the same time. Therefore, they can't move anything. Okay? Other forms of damage to one or more of these basal nuclei show up as unwanted movement, in the sense of almost athetoid or sinuous movement. So they'll sit there and it'll just move constantly, and you wonder how in the world do they ever get any rest. They'll sit on a hand and try to stop it, but it'll eventually find its way out. When they fall asleep, those movements stop. Okay? Huntington's chorea is a form of this. It's a genetic disorder that's inherited. And a few years ago, I had a student whose grandfather she'd taken care of had died of Huntington's chorea. Her two uncles, who were in their early 50s, had it. And her mother, they went annually to Huntington chorea conferences, and her mother had decided to be tested for it. And her mother had it. And was just starting, she was in her early 40s, and was just starting to show signs of it. But my student wasn't ready yet to find out, and so she elected not to be tested for it. She decided she'd rather believe that she didn't have it rather than to know that in 10 or 20 years she was going to have those symptoms. And we discussed it several times, and she said, you know, it's really hard when I'm dating to decide when is the proper time to tell a boyfriend, you know, that I have this, you know, if it's just a casual friend, or if it progresses, you know, it's kind of your time to decide, you know, if you had a terminal cancer, when you tell somebody, hey, by the way, we're falling in love with each other, but I'm not going to be here, so, you know, do you scare them away or not? And so I mean, she was planning her life. She was going, you know, wanting to become an ultrasound tech. and so on. But I'll show you some videos when I get my PowerPoint thing, the projector to work again, of some of these particular movements, okay? But they're involved in this area. Another is, they'll test for affecting this is to have, I can turn my hand rapidly, go back and forth like that, okay? Some individuals with problems with parts of their basal nuclei have trouble starting and stopping. They'll hesitate, or they'll just do the same thing again instead of flipping back and forth, okay? So it's not the actual upper motor neuron to spinal cord lower motor neuron pathway that's affected, it's the modulation of those movements. With Parkinson's disease, there is a constant tremor, okay? Sometimes a pill rolling type of a tremor. And this gets better when the person has an intentional movement. So Muhammad Ali lit the flame in the '96 Olympics in Atlanta, and he was holding the torch as he moved to light the flame for the event, and his hands were shaking so much you wondered how he could even, get. But as he went to actually light the flame, that shaking almost disappeared. So the intentional tremors disappear with intention. When we talk about cerebellum on Monday, those tremors get worse. The harder you try to do a movement, the worse the tremor, this moving back and forth becomes. And we'll talk about the cause for that. Okay, let's stop here and take a break, and then we'll come back for just a short discussion in the diencephalon of the thalamus, and we'll leave hypothalamus. We're actually kind of in between today and tomorrow's lecture, and we'll leave hypothalamus for tomorrow. Hopefully the PowerPoint